And Hans group really has been one of the great pioneers in developing computational methods in analyzing medical imaging data with the focus on outcome. And one of the things that really kind of highlights from his work is actually making sure that things work. You know, when you actually put them in the hands of the clinicians, making sure that they are robust to different scans. I mean, when you go to the clinic, you have things from different vendors, different clinicians play things differently, and you need to make sure that from a matter point of view, right, to when it gets to the hands of the clinician, things actually work, because you're not always there to fix things. So, with this introduction, I would like to welcome Anant. Thank you for coming here, and please, we are looking forward to talk.
Thanks, Pirang. Very happy to be here. Of course, after that introduction, it's going to be all downhill, because obviously I can't live up to all that hype. But I'm very happy to be here. Really enjoyed the last day and a half in Vancouver. You have a beautiful city, beautiful campus. So hopefully I can provide a little bit of insight into the kind of work that we do, and there's going to probably be a fair number of things I talk about, and I'll try to leave some questions at the end where we can sort of brainstorm and discuss. I'm going for breadth here rather than depth. When I was trying to prepare the talk, I was sort of juggling in my mind, you know, this is mostly electrical engineers and computer engineers. Should I focus on the methods? Should I focus on the use case? And finally, I decided, let me just try to show pretty pictures and try to convey the ideas rather than the underlying methods or the underlying algorithms, which of course, I'm happy to direct you to the specific papers and reports and so on. But my goal here is really to showcase, you know, how computational imaging methods could be brought to bear on medical image data, in fact, all types of data, and how we can prize out patterns and signatures from that data to be able to predict the risk of disease, aggressiveness of the disease, outcome for patients, and potentially even trying to predict what kind of therapies might work, all based on computational image analytic methods that we can bring to bear on data and then try to glean information. And I use the term here subvisual because, you know, the idea here is to be able to go in and capture information that will most likely escape visual examination, visual interpretation by a human. And hopefully what I will try to convey is the fact that there's a lot of rich information with the right tools one can unlock. And one can now find correlations between this subvisual image data, embedded data, and outcome and disease risk and disease aggressiveness. I should also say, and Pirang just mentioned, we've had to switch at the very last minute from my PC to a Mac, so some of the movies, unfortunately, are not going to play, but I'll try to labor through the slides the best I can. Just a few conflicts of interest. I have stock holdings in Ibris Inc., Elucid Bioimaging Inc., and Diagnostic Precision Inc. I'm also a scientific advisory board member and consultant to Diagnostic Precision, and I have a couple of NIH academic industrial partnerships, one with Siemens and one with GE. So big picture. Really, we know for a fact that there are different types of data that are being routinely acquired for disease characterization. Everything from imaging down to pathology, down to molecular data. And the question a number of groups are asking, and ours is just one of several groups, which is how do we try to encapsulate, embed, exploit all of this information that's spread across different length scales. You've got radiographic data that resides at sort of a macroscopic length scale. You've got pathologic information that gives us disease characterization at a cellular scale. And then you've got molecular information that literally gives you information at the protein level, the gene expression level. All of these different types of data, these different channels, are characterizing the disease in different ways, providing different lenses with which to view the same disease. And therefore, they're complementary. And the big question is, how can we potentially exploit these different pieces of information, bring them together to be able to create better predictors for disease diagnosis and prognosis? So I use here as an example prostate, which this group has a great deal of experience and expertise in. So what you're seeing there is a region of adenocarcinoma outlined in the white box on a T2-weighted MRI. And of course, with a multiparametric exam, one can start to interrogate the data using different parameters. So you can use ADC, T1 with contrast, MR spectroscopy to look at choline and creatine and citrate ratios. So you've got a multiparametric characterization of the macroscopic length scale. Now, assuming this patient goes on to have a radical prostatectomy, which means that the prostate is physically excised, you can look at that same region of cancer that I showed you on the MRI, and now with computational analytic tools start to look at cellular characterization, cellular architecture, look at tissue texture in a far more granular way, and start to glean different measurements, different pieces of information, different pieces of data from the pathology, from the histopathology that is not appreciable on the imaging, but provides complementary information. And then, of course, one can look at the pathology data with finer resolution, with greater resolution. You can start to look at nuclear architecture. You can look at nuclear shape, nuclear morphology. And then one can scrape off that tissue within the white box I showed you on the T2 weighted MRI and start to do omic analysis. In this case, I'm showing you protein expression data acquired via mass spectrometry, where that same region of cancer has been scraped off and passed through a mass spectrometry machine to give us peptide expression. So you've got different, an idea of the kinds of proteins that are expressing, overexpressing, or underexpressing within that region of disease. So we've got information across these different length scales, macro to cellular down to the molecular level. And we want to try to be able to fuse this information to create better predictors of diagnosis and prognosis. So from an engineering perspective, from a computational perspective, we've identified some very significant challenges to be able to do this multi-scale, multimodal data fusion. So one of the first issues, and this is, it was very, very nice to see all the wonderful work going on in this group at UBC in the context of co-registration. So knowledge alignment, which is bringing the different pieces of information, the radiology, the pathology, the molecular data, is clearly is a major challenge. So this is only one of the critical challenges that have to be addressed. The second challenge is, of course, knowledge extraction. How do we convert all of these different pieces of information into quantitative measurements, into quantitative descriptors, so we can, in a robust way, quantitatively describe the disease appearance, and do this across different length scales. And then finally, the most perhaps subtle challenge of all, which is knowledge representation across different modalities and different length scales. So if you think about it, the MRI data gives you a handful of features. The pathology data, as I will show you, could potentially yield hundreds of features. And the molecular data, of course, could yield, based on the number of proteins or genes you're looking at, tens of thousands of features. So what you've got now is this issue of dimensionality mismatch. You've got challenge of reconciling dozens of features with hundreds of features with thousands of features across different length scales. So what are the kind of computational challenges to be able to represent that knowledge across different length scales and create fused predictors that is appreciative of the fact that information resides in different dimensions, in different modalities, in different length scales? For my talk, I'm really going to focus more on the knowledge extraction piece and a little bit on the knowledge fusion. I'll briefly touch on the knowledge alignment, but again, like I said, I think there's a lot of expertise in this room, perhaps even more than the expertise in my own group on co-registration knowledge alignment. So I'm going to sort of skim on the details on the first challenge and really focus on the latter two challenges. So let's talk about subvisual image features. And like I said, the challenge is being able to go in and extract information that may not be visually appreciable by a human, but still may be prognostic or diagnostic, or may be correlated with disease outcome. And there is an opportunity now to be able to bring to bear, with sophisticated pattern recognition, image processing, or as the flavor du jour term, which is big data analytics, or big data, right? Everything is big data. So can we bring to bear these big data analytic tools on this image information across different length scales and different modes to be able to tease out the subvisual encoded information, embedded information, and try to find links between outcome, prognosis, disease risk, and these subvisual image features? And we can start to do that because of the fact that we have far more sophisticated tools and arsenal than we've ever had before, and also because we have now the hardware that allows us to get more grandiose with our methods and with our analytics. So what are these examples of the subvisual features? Well, one obvious example is texture, and I'm sure a lot of you in the audience are aware of what texture is. But sort of very loosely speaking at a high level, it refers broadly to different ways in which we can capture spatial heterogeneity, different ways in which we can capture subtle patterns in the image via a series of different mathematical operations. And there are different ways in which you can capture these local intensity statistics. Of course, one can look at the so-called first-order statistical features, where you're looking largely at just mean and standard deviation within small neighborhoods, or you can start to look at higher-order statistics, such as co-occurrence statistics, and look at the co-occurrence of and proximity of different image intensity values. Or you can look at steerable filters, where you can actually look at gradient orientations in particular directions, such as with the Gabor filter bank, and capture these subvisual image features. So there are multiple different ways in which these subvisual texture features can be garnered. And subvisual texture features, of course, have been used a great deal over the last 2 decades at least, in the context of medical image analysis and radiographic image analysis. And in fact, we made a small contribution in this direction recently, the McKay meeting, where we presented a way of capturing localized orientations. Rather than looking at a global orientation filter such as the Gabor filter, we focused more on trying to understand at a local level what are the interplays between gradient orientations. So do gradient orientations tend to line up? Are they more ordered or disordered in certain pathologies at a local level rather than just a global level? And we call this the collage texture feature, and I'll talk a little bit about it in a couple of slides. But the point here is, of course, that these subvisual representations that I'm showing you at the radiologic scale can really tease out information that may not be visually apparent, but could tell you a great deal about the outcome, about the risk, and potentially about the biology of the tumor. And that sort of segues into this whole notion of radiogenomics, which is, can these subvisual image features from radiographic data potentially inform us about the molecular status of a number of diseases? And we've been looking at breast cancer as one particular use case, where we've been very interested in trying to look at MRI information, or breast MRI data in particular, and see whether features that we're getting from the breast MRI could tell us about the molecular status of the tumor. So the molecular status of these tumors is typically ascertained after biopsy. So a biopsy would be done, you do an immunohistochemistry test, you look at HER2, estrogen, progesterone, and based on the levels of expression of HER2, the HER2 gene, estrogen, and progesterone receptors, you make a diagnosis of whether this is a ER-positive breast cancer or a HER2-positive breast cancer. Or, of course, if there's no expression of ER, PR, and HER2, then you classify it as a triple-negative cancer. And triple negatives are the most virulent, the most aggressive type of breast cancer. But all of that happens following the tissue excision, following the obtaining of the biopsy sample. When the immunohistochemistry is done and the FISH test is done, that's when you get that information. So the question we were asking is, if you look at an MRI prior to a biopsy, is there a way that we could predict what the molecular status might be? And that could provide a way of quickly identifying which ones are the more aggressive cancers, which ones do we need to get to and potentially start to treat as soon as possible. So what we did in this paper that we published in Radiology earlier this year was actually show that texture, dynamic texture, or changes in the texture of the lesion during a T1 contrast exam actually appears to have very different signatures between different molecular subtypes. So people have been looking at dynamic contrast enhanced MRI exams for breast cancer for at least 3 decades now. We took a slightly different approach. Rather than simply looking at the contrast wash-in or wash-out within the lesion, we looked at texture changes within the lesion over a period of time. And based on how the texture changed, we realized, we found that we could actually separate out these molecular subtypes with a fairly high degree of accuracy. In particular, what was very interesting is we were able to separate out the triple negatives from other subtypes with an AUC of about 0.97. And this was a relatively small study. I mean, this was about 79 breast cancers we looked at. So it wasn't powered by any means. It wasn't, we didn't have sort of an independent validation set, but it certainly provides the proof of concept. It provides the preliminary data to suggest that there are subvisual image cues that can be extracted via computational image analysis that could tell us about the biology of these tumors in a non-invasive way. I mentioned the collage feature that we looked at recently, and a little bit more about this particular feature, something that I'm very excited about, where we've been looking at local orientations, local gradient orientations. And what we found is when we look at these different types of tumors, there is some very distinct patterns that emerge at a local scale that are not appreciable at a global scale. In particular, when we looked at these gradient orientations at a local scale, we found that in certain kinds of tumors, the gradient orientations seem to be more chaotically arranged as opposed to our subtypes. And the nice thing is there is actually, from the co-occurrence texture feature approaches and Harlick feature approaches, there actually is a pretty nice mathematical framework to be able to express the disorder in these gradient orientations. So we actually compute the entropy of these local gradient orientations, and entropy actually captures the extent of disorder or order in the gradient orientations. So in fact, if you see a high expression as seen in those hotspots within the lesion, if you see a high degree of entropy, it means there's significant disorder, significant chaos within these local neighborhoods. And if there's actually low entropy, it basically suggests that those orientations are more organized, that there's less chaos and more order in those orientations, which actually is a good thing. And if you think about all the literature that's now coming out about tumor heterogeneity, particularly intratumoral heterogeneity and disease risk, a lot of papers now suggesting that the more heterogeneous a tumor, the higher its risk, the higher its aggressiveness. So we've been very excited because this collage feature, these local gradient orientations, suggest a potential way, one way, not the only way, but one potential way of characterizing intratumoral heterogeneity on imaging that might allow us to assess disease risk in a non-invasive fashion. Because if you see a lot of high entropy expressing areas within the lesion, it might suggest there's a lot of disorder, a lot of chaos within the lesion, which in turn suggests high intratumoral heterogeneity, which in turn suggests that this is a high risk, highly aggressive tumor. And we looked at the same cohort that we published in radiology in this McKay paper that we presented in September in Boston. And the collage expression actually was significantly different for the triple negative tumors that I mentioned were the more aggressive phenotype versus the less aggressive phenotypes such as the ER positive or the fibroadenomas, for instance. A large part of what we've also been doing has been on the pathology, and I sort of start off by talking about some of the histopathology features. And the notion of computational image analysis, the notion of subvisual features is not certainly limited radiographic data alone. In fact, I think the implications for pathology are even more immense. To the point where I think you can almost think of the quantitative histomorphometry or the subvisual image analysis on pathology image data as sort of a digital stain. So we actually now have, with the right tools, a way of overlaying on the pathology images a new mathematical representation of the architecture of the morphology of the tumor that doesn't actually require a destruction of the tissue. So any immunohistochemistry test, any protein marker test involves the destruction of the tissue. But what if you could take a tissue image and apply the right analytic tools to the tissue image and come up with a morphology map or histomorphometry map that characterizes nuclear architecture and nuclear morphology? And this is the kind of images you get, where the left panel essentially shows the original H&E hematoxylin eosin stained image. The center panel represents a graphical a network that captures the cellular arrangement of individual nuclei, and to the right here represents the textural map or the textual representation of that image. So you've got essentially now a digital way of staining this image where we haven't touched the tissue itself. We've just taken an image, a digital image of the tissue, and come up with a way of representing all that very, very embedded, deep, big data in a quantifiable manner, in a way that no pathologist can possibly digest or possibly interpret. But the right analytic tools can allow us to try to extract more information than can be visually discerned. And the beautiful thing about this is, of course, you can start to look at this nuclear architecture across different fields of view, and you can look at nuclear texture across fields of view, because that's with the right tools, with the right hardware, with the right processing power, you can essentially scan the entire slide, the entire digital image. And you can see here that as you start to increase the field of view and the resolution, you start to get more and more complex networks, and the texture starts to get more and more heterogeneous. But you can keep going, and you can start to look at larger fields of view. The graphs become more immense and more complicated, and that's sort of both the opportunity as well as the challenge. You've got very, very complex networks representing very, very high orders of information, high orders of architectural arrangement between these different cells. But then you also have the challenge of how to be able to make sense of this, how to interpret all of this, and how do we connect this now ultimately to patient risk, patient outcome, and patient treatment. So that essentially begs the question, therefore, can the quantitative histomorphometric analysis of tumors allow us to predict disease aggressiveness and patient outcome? And obviously, as you can guess, the answer, we think, is yes. That we can now look at digital images of these tissue samples and be able to tell which patients have more aggressive disease, which patients are going to respond to a particular therapy or not, and what might be the appropriate therapy to apply to a patient, all just based of computational analytic tools that are being brought to bear on routine pathology data, or routine image data for that matter. So all of these features that I showed you actually can translate, these graphical networks that I showed you, can translate into a series of quantitative measures. And I'm just throwing up this table here. I don't intend for you to digest all of the data that's thrown on here, but just want to show you some of the examples of the kind of features that you can mine from these graphical networks, from the graphical arrangements, and how these features now can allow us to build predictors that tell us who is going to respond to a particular therapy, what kind of disease response is going to occur, and so on. But let's get into some use cases. I mean, hopefully what I've done so far is set the stage for some use cases and how we can bring to bear these tools on particular problems. And I want to start with some of the work that we began about 6 years ago as a conversation between a breast oncologist at the Cancer Institute of New Jersey and myself. And he was looking at some of the work I'd done, and he said, you know what, I've got a great use case for you. I want to talk about breast cancers. He's a breast oncologist. And he said, the big problem with breast cancers, in particular ER-positive breast cancers, is that we don't have a definitive way of knowing how to treat these patients. The problem is most of these ER-positive breast cancers, the estrogen receptor-positive breast cancers, tend to do very well. That means that if you use standard hormonal therapy alone, which is the first line of defense to anyone with ER-positive breast cancer, most of these breast cancers are going to do very well. It's only a small minority of these breast cancers that require chemotherapy. Chemotherapy, as you can imagine, is more aggressive. It results, obviously in hair loss. It has all of these deleterious complications that we're all very well aware of. So how can we avoid a brute force approach? How can we avoid an approach where we don't throw the kitchen sink at everyone who has ER-positive breast cancer? Can we avoid and selectively identify those patients who will benefit from the chemotherapy, but then avoid chemotherapy in patients who will do well with just hormonal therapy alone? So he threw this problem at me, and that got me thinking. But then it turned out there was already somebody in the market who offered a product. And this is a product offered by Genomic Health. In fact, now there are a couple of other products out in the market as well. But all of them are based on the assumption that the information, the prognostic information, is embedded in the genes. So Oncotype DX, which is a product marketed by Genomic Health, essentially takes the tissue, it mashes it up, it destroys the tissue, and looks at the gene expression of certain genes. And some of those are listed here. And comes up with a classifier that looks at the expression of these genes and then gives a risk score. So if you get a low risk score between 0 and 17, it means that you are a good candidate for hormonal therapy alone, ergo you don't need chemotherapy. If you have a score from 30 onwards and beyond, then it means that you probably are a candidate for chemotherapy. If you're somewhere in between, it means, well, you probably don't, the test doesn't know. I mean, essentially, you have the oncologist makes the final call. So this test existed, but my breast oncologist colleague basically challenged me and said, well, you know what, I sense there's information in the pathology already. Can we apply your tools? Can we go in and start to look at these breast cancer specimens and see if we can predict which of these breast cancers are going to have good outcome versus poor outcome? In other words, which of these breast cancers would require chemotherapy and which ones would not require chemotherapy? And could you just do it off an analysis of a digital image? And that so gave rise to the idea of the IBRIS, or the image-based risk score. The idea was very simple. You take a slide, a tissue slide, and any woman who is suspected of breast cancer, let alone has breast cancer, is going to have a tissue slide. It's going to have a biopsy slide. That biopsy slide can be scanned using a routine whole slide digital scanner. You get a high resolution digital image. We apply our analytic tools to look at nuclear architecture, nuclear morphology, and come up with a prediction of is this aggressive disease or less aggressive disease, and therefore now can we figure out whether this is a candidate for chemotherapy or hormonal therapy alone. So that was the basic idea of IBRIS. And you can appreciate immediately, the nice thing here is that it's non-disruptive. It doesn't disrupt the clinical workflow in any way. The slide is not being destroyed. The slide is already present. That's already going to be created. And all you need is an image. And the image can be created using the slide scanner. And you guys have one here at UBC. Most sites, most centers now have whole slide digital scanners. So there's no biggie there. I mean, it's essentially just a fancy high-end scanner to the ones that have been around for a couple of decades now. Only thing is that these scanners scan slides. But before we could actually go and start to ask the question about aggressiveness or signatures or markers for aggressive disease, we first had to figure out where the cancer was. So we basically started to get interested in this notion of deep learning, and I'm sure a number of you are familiar with this idea, which sort of has been around for a few decades now. I mean, we've all heard about neural networks. But I guess deep learning got its big boost a few years ago with the paper that came out from Andrew Ng's group from Stanford, which looked at YouTube videos to try to figure out whether cats were present in YouTube videos or not. And they got a certain level of accuracy in figuring out which YouTube videos had cats and which ones did not. So we said, okay, well, can we bring that idea to bear now on pathology images? Could we build deep learning models to be able to figure out where cancer was on images? And the reason these histopathology images make good candidates for deep learning is because they're so big. Because a single biopsy core that's scanned on a digital slide scanner turns out to be about 2 gigs of data. So that's 2 gigs of data from a single pathology slide, a single biopsy core. So from a deep learning perspective, from a big data perspective, it's the ideal use case. Because there's so much data that's available. In any case, we've got to figure out where to interrogate, where to do the analysis, where to go in and look for our features. So Angel Cruz, who was a visiting student from Columbia, National University of Columbia down in South America, spent a summer last year with me and programmed a deep learning network to find invasive ductal carcinoma on pathology slides. And I won't go into the details, but I'm happy to send you the papers if you're interested. But essentially, if you looked at a manual annotation from a pathologist, the deep learning model is able to very quickly identify exactly where the cancer is, and in fact, if anything, becomes even more intelligent than a pathologist, or well, perhaps not more intelligent, but certainly more accurate than a pathologist, because it's able to, in fact, leave out the interleaved stroma and is able to focus on the tumor epithelial regions alone. So, in fact, those little gaps there are not actually false and negative errors. It's just that the model gets so intelligent that it can actually tease out exactly where the stroma is, where the fat is, and focus on the tumor epithelial regions. And so we get very, very precise maps now of exactly where the cancer regions are. But then we've got all of this big data problem. We've got all these features that we've extracted from regions that we've identified as cancer. We've got to make sense of all of this. So Ajay Baswanhali, my former graduate student and postdoc, essentially developed a multi-field of view classifier that we published last year, where essentially the features could be extracted across different fields of view, across different resolutions. And he was able to create a boosted classifier that combined the features across different fields of view to come up with a risk prediction model. So to validate this model, we looked at a series of breast cancer patients, ER-positive breast cancer patients, all of whom had biopsy samples. So the tissue slides existed for all of them. For all of them, we scanned the slides and got high-resolution images. But what we also had for all of these patients was the oncotype score. So the oncotype, again, is the gene expression score that we have from Genomic Health. So all of these patients had that score. And we also knew whether it was a low score, an intermediate score, or a high score. What we did was we extracted the features using the multi-resolution model, using the graph-based features, the texture-based features that I talked about. We did it for all of these 160 patients, and then we mapped the data into reduced dimensional space. So we had a very high dimensional feature space. We used projection methods to map the data down into a low dimensional space. So that's what you see there. And then using nonlinear dimensionality methods, we unraveled that manifold into a linear scale. So we mapped it out onto a single line because we wanted to get to a risk score just like Genomic Health's Oncotype DX. So we wanted to have that sort of analogy with the gene expression based test. What we also did, once we'd done the mapping, now we said, okay, now let's go and unravel the Oncotype DX scores. So we know that the Oncotype score can be low, which is good, intermediate, which is I don't know, and high, which is of course more aggressive. So we said, okay, let's give different colors. Let's give a blue for the low scores, green for the intermediates, and red for the aggressive ones, and that's the result we get. So what is really interesting is we have just done a mapping of the data based off the image-based features, the histologic image features. We mapped it onto a line, but when we unravel the oncotype scores, you can see there's a very, very nice concordance in this data set between the oncotype, between the gene expression test, and the histologic image-based risk score. In other words, the image-based features from the tissue slides seem to concur, seem to agree very nicely with the gene expression-based predictions. So this was very, very heartening. This was very satisfying. But immediately the question came up when I showed this data is, well, that doesn't mean anything. It doesn't mean anything because the oncotype score is only a surrogate. It is not ground truth. It is another test. It's another assay. All you've done at this point is shown that your assay, your image-based risk score, agrees with another assay. But that assay is a surrogate. Ultimately, the ground truth is outcome. Ultimately, the gold standard is outcome. So that forced us to think about this. So we said, okay, well, clearly we need to go after outcome data. We need to go find patients where we know the long-term outcome. We need to look at those patients and see whether the image-based risk score can stratify outcome. So thankfully, because of my collaborator, my breast cancer oncologist collaborator, Dr. Ganesan, we were able to get hold of a small cohort, 72 patients, for whom we had outcome. That is, we had information about distal recurrence of breast cancer versus early recurrence. And what you're seeing here are the Kaplan-Meier curves. So these are survival curves that essentially we're generating from the image-based risk score. The image-based risk score was applied to stratifying outcome in these tumors, some of whom had favorable outcomes, some of whom had poor outcome. And the separation between the two curves essentially suggests that the image-based risk score was able to distinguish patients who had early recurrence of breast cancer versus distal recurrence of breast cancer. In other words, good outcome versus poor outcome. And the differences were statistically significant. So this is yet unpublished, but this has just been accepted at the USCAP meeting, the United States College of Anatomic Pathologists, the United States and Canadian Anatomic Pathology meeting that will be presented in March next year. So this seems like a step in the right direction. Clearly, we need to validate this on hundreds of patients, potentially even thousands of patients. But again, it's a step in the right direction, because not only are we showing concordance with the gene expression-based test, independently, the histologic image-based risk score seems to be able to stratify outcome as well. So why is this important? Why is this important? And I just want to this is my tongue-in-cheek comparison between Oncotype and IBRIS. So this is really meant to be tongue-in-cheek. It's not an endorsement. It's not an advertisement for image-based risk score. But I really want to drive home the message about how, particularly in the developing world, particularly in the lower middle income countries, where a image-based predictor that does not destroy tissue, that uses just digital images, can really have a major impact. It can have a major impact because the Oncotype DX test is $4,000. That means in lower middle income countries, you simply cannot do this test. It's just way out of reach for people who would need this test. The image-based risk score, that $400 is just an arbitrary number. It could be $40, it could be $100. The point is that the cost of goods sold via an image-based risk score is minimal. You just require an image of the tissue slide and you're good to go. The results, of course, could come back within hours. With the Oncotype, you have to physically ship the tissue out to California, and it's at least a 2-week lag before you get the data. The tissue specimens are destroyed with Oncotype. With IBRIS, of course, they're digitized, so you have the images for posterity. Accessibility is largely limited with Oncotype to US only. With IBRIS, of course, it's available worldwide. So in other words, you have an inexpensive, fast, reliable, accessible prognostic breast cancer test that's priceless. For everything else, there's MasterCard. So what else can we do with the image-based risk score? So one of the other experiments that we're looking at right now is looking at pre-malignancies. So cancers that are not yet cancers per se, but are in their pre-malignant stage. One example is ductal carcinoma in situ. So it's called a stage 0 breast cancer, where it's still, it's not become invasive yet. It's still circumscribed. It's still defined as what is called in situ disease. So one of the big challenges here is trying to figure out which of the DCIS will actually progress on to full-blown cancer and which ones will not. So we've been looking at this problem, again, using our image-based features and trying to figure out, if you look at a DCIS that progresses on to invasive cancer and ones that don't, can we distinguish between the two? And our very, very preliminary data on about 23 cases seems to suggest that, there is some separation. I mean, I think we clearly need to investigate this even further. But again, there is some reason to be cautiously optimistic that there are features there that might be able to tell us which pre-malignancies move on to an invasive condition and which ones do not. We've also had recently, over the last couple of years, started to look at head and neck tumors. And in head and neck tumors, and I'm sort of going to jump between pathology and radiology, like I suggested in my very first slide. So I'm sort of switching back to radiology here. But one of the big challenges in brain tumors is trying to figure out response, or treatment response. And one of the challenges in brain tumors is that after treating brain tumors, in a few months, very often on MRI, there is a change in signal in the pattern on the region that's been treated for brain tumors. And very often a neuroradiologist is unable to tell whether what you're seeing on the MRI is the tumor returning, has the tumor come back, or is it just an effect of radiation therapy? Is it just so-called radiation necrosis, which is a largely benign condition? So we, in particular, Pallavi Tiwari, who's a research faculty in my group, has been taking a crack at this problem. And what Pallavi has been doing is to try to look at some of these subvisual features, the collage feature, to see whether she can actually distinguish between radiation necrosis and recurrent brain tumors. We actually looked at now 48 cases of recurrent tumors as well as radiation necrosis. And what was very, very interesting is when we looked at these gradients, when we looked at these subvisual image gradients and quantified it using that entropic measure I talked about, the entropic expression actually was significantly different between radiation necrosis, and recurrent tumors. In other words, the recurrent tumors seem to have a higher entropy, and they appear to be way more chaotic compared to the radiation necrosis. The radiation necrosis, the entropy in general seemed to be much lower. So even though the signal intensity looked very, very similar, the subvisual features seem to have a very clear and distinct separation. One of the other things that Pallavi has been looking at has been to try to see whether off a baseline MRI, whether we can predict longer-term outcome for these patients. So GBMs, or glioblastomas, are really the most aggressive, virulent type of brain tumors there are. And if in brain tumors, when you talk about long-term outcome and short-term, or rather long-term survival and short-term survival, it's very, very relative. I mean, really, we're talking about difference between 9-month, 6-month survival and 3-year survival at most, right? So that's sort of longer-term survival and shorter-term survival. But the question we're asking here is, can we see features on a baseline MRI with subvisual image analysis that could distinguish between longer-term survival and shorter-term survival? And this is data that we just presented at the BMES meeting, as well as the Society for Neuro-Oncology just 3 weeks ago, where we showed that these collage features, again, could separate out patients who had favorable survival, again, relatively speaking, from those who did not have favorable survival. And if you look at the signal intensity alone, those p-values were not statistically significant, but with the collage features, we see statistically significant difference in the overall survival for these patients. So just a quick point about these Kaplan-Meier curves, or interpretation of these survival curves. The greater the separation, the better your classifier, the better your assay. So you can see sort of a nice separation between the favorable survival and the unfavorable survival. I want to skip back to pathology now. This is a NCI-funded project where we're looking at oropharyngeal tumors, or head and neck tumors, that traditionally have favorable outcome. These are so-called P16 positive oropharyngeal tumors. The P16 is a protein that essentially tells you about the HPV status, the human papilloma virus. So these are tumors that are caused because of the human papilloma virus. And HPV tumors in general tend to have favorable outcomes. So because they're virus-induced, they tend to not be as bad. So most of these patients, by and large, have pretty good outcome. But even within these tumors, there are those that don't do that well. So about 75–80% of these tumors tend to do very well with standard treatment, but in about 20% of the cases, these tumors will have metastasis, will have progression. So the question we asked here was, again, are there features that we can glean from the image data that could tell us about which patients were going to have good prognosis, which patients were going to metastasize and have progression? So we took a slightly different approach here. We took our nuclear architectural approach, but we decided to actually separate out the stromal compartments from the epithelial compartments. So the stromal compartments essentially is the region within the tissue that is sort of the more uninteresting piece. So the tumor epithelium is really where the action is happening, because everyone's interested in the tumor compartment. You're looking at the tumor cells and tumor morphology. But more and more, there's been interest recently in looking at the stromal compartment. So the stroma is sort of the more uninteresting part of the tissue. It's where you don't have too many cells. It tends to be fairly bland, mostly just cytoplasm there. So what we said was, let's construct a graph network separately for the stromal compartments as well as the tumor epithelial compartments. So that sort of gave us the idea of SPACLE. I mean, this is essentially an acronym for our approach, where we created graphs within the tumor epithelium, within the stroma, and combined the features from those two different partitions and created a classifier. And the idea was, with this classifier, to see whether we could separate out oropharyngeal tumors that had metastasis from those that did not. And our data, which we published in the American Journal of Surgical Pathology a few months ago, was very, very exciting. I mean, because you see that leftmost panel there, the SPACLE predictor essentially split apart the patients that did not progress from the patients that progressed very, very nicely with a highly statistically significant p-value. The center and the right panels represent the Kaplan-Meier curves we got from the pathologist. So the pathologists were looking at essentially two variables, lymph node status and looked at stage, tumor stage. But when they looked at those two variables and we looked at the corresponding Kaplan-Meier curves, they actually were not as clearly discriminating as the spackle features. So this was very exciting on two fronts. One was the image-based features could actually separate out the tumors. But what was even more exciting was they were able to do it better than pathologic variables. So I talked about the subvisual embedded data. And this is a classic example of where the subvisual embedded data did do better than what the pathologist was visually seeing on the tissue slides alone. I have to talk about prostate cancer because it's an area that I've been working on for about 10 or 11 years now. And of course, UBC, you've got some of the world's experts in prostate cancer, in Tim and Purang and Rob. So I have to talk about some of the work we're doing. And our focus has been more on the prognostic side and more on how to bring in the pathology piece along with the imaging to try to inform the different scales and the different modes of data. So, like I said, right in the very first slide, our focus has been sort of on this correlation of information, the fusion of information across different length scales and different modes, and how can one piece of information inform the other mode or the other length scale. So this is an example where we looked at correlating and combining information from pathology with in vivo MRI data to be able to figure out more sensitive, more specific predictors of aggressive prostate cancer in vivo. So we decided to focus on patients who were getting a radical prostatectomy, who had a multiparametric MRI exam. Once the prostates had been removed, we did an immunohistochemistry. So we went in with a protein, in this particular case CD34, which essentially stains for microvessels on the pathology specimen. And why that's nice is because you can look now at the microvessels on the pathology and create a graph network that looks at the arrangement of the microvessels. And it turns out, very interestingly, that more aggressive tumors tend to have a different arrangement of microvessels compared to less aggressive prostate cancer. So we said, okay, we can establish that on the pathology because we have the tissue already, because it's come out. Now can we correlate it back to the imaging that was done before the surgery and learn from the MRI what are the features that are associated with more aggressive prostate cancer and what are the features associated with less aggressive prostate cancer. So a number of things had to happen in order to make this possible. One is the co-registration, and we've done work on fusion of pathology and MRI, ex vivo pathology and in vivo MRI. So first thing to do was to fuse ex vivo pathology with in vivo MRI, map the extent of tumor from the ex vivo pathology onto the in vivo imaging. We extracted a series of features from the DCE MRI in this particular case, as well as the architectural features that characterize the microvessel architecture. And now did this sort of radiohistomorphometric plot, where on one axis you've got all the immunohistochemistry histomorphometric features. On the y-axis you have the radiologic features, or maybe it's vice versa. Sorry, it's radiologic features on the x-axis and histomorphometric features on the y-axis. And we did a correlation analysis and identified the features that strongly co-expressed. So what DC MRI features strongly co-expressed with the histomorphometric features. And then in an independent set, we said, if we look at those MRI features that were co-expressing with the histomorphometric features, can those separate out between high-grade tumors and low-grade tumors? And the results were very, very exciting. And we actually have a paper under review in JMRI right now, where we actually saw three clusters emerge. We saw one cluster where primary grade 3 tumors were present. This is a clustering just based off the DC MRI features alone. We got one cluster where it was just the Gleason grade 3 tumors, primary Gleason grade 3 tumors. We had another cluster where we had 4 plus 3 tumors, so primary grade 4 and secondary grade 3, and another cluster which comprised of 4 plus 4, so primary grade 4 and secondary grade 4. So I haven't really explained what Gleason grade is, but it's really the most important predictor of aggressiveness and outcome in prostate cancers that we know of today. And really, a Gleason grade 3 is a rather indolent disease. Anything with 4 and above is something that one has to take more seriously. 4+4 is high risk. It's something that has poor outcome. And a 4+3 is somewhat more intermediate. So we, using this sort of correlation of the histomorphometry and the radiology, found that we could actually distinguish between sort of the more indolent, the lower risk, the intermediate risk, and the high risk, and sort of all mapped out quite nicely into these 3 different groups. And that's a very, very small n, and I totally understand that, and we have to do a larger independent validation, more rigorous validation. But it suggests that this sort of convergence of pathology and imaging could really inform on the image data and could enrich the image data. So we've been sort of working on this, and unfortunately this movie will not play, but this is work that one of my postdocs, Mirabella Russo, has been working on as part of a postdoctoral DoD fellowship. And what Mirabella has been doing has been to look at patient populations. So she's been looking at patients who get a radical prostatectomy and looked at the corresponding imaging data and mapped all of that into a single canonical frame, into a single atlas representation. And why this is useful is because it allows us to go in now and appreciate the spatial distribution of the disease in different parts of the prostate in one unified canonical representation. And she's extended this idea to now looking at patients who have favorable outcome or unfavorable outcome after surgery. In particular, patients who have biochemical recurrence following surgery from patients who do not have biochemical recurrence. So we're actually working with Ash Tiwari's group in Mount Sinai. We got about 72 patients for whom we had outcomes. So we had patients who we knew had PSA failure or biochemical recurrence within 5 years of surgery from patients who did not. So what Mirabella did was to create 2 atlases. She created one atlas for patients who had biochemical recurrence within 5 years and another atlas for patients who did not have biochemical recurrence. And then what she proceeded to do was to create a difference atlas, a differential atlas. And again, I apologize because this movie will not play. But the punchline was that she found when she did a differential between the 2 atlases where there were unique shape-based differences on the capsule and within the central gland of the prostate between the 2 cohorts. And this was an amazing discovery because nobody has ever looked at this or thought about this or identified this. So in some senses, it was sort of finding by chance that patients who go on to have poor outcome appear to have a differently shaped prostate than patients who have good outcome. And we still don't know exactly why this is the case, but over 72 patients this seems to hold, and the values seem to be statistically significant as well. But the thought is that it may be that in more aggressive disease, the tumor is pushing out. The tumor is pushing out. These are cases where you typically tend to have extracapsular spread. And because the tumor is pushing out more aggressively, it tends to distend the capsule in a particular way that's unique and different from patients where it's more slow growing, where it's more indolent. And that may explain the reasons for the differential shape between the 2 populations. But I want to come back to pathology. Like I said, I'm sort of flitting back and forth between radiology and pathology. So I want to come back to pathology now. And show you an example of signatures or features that we've been looking at to try to distinguish between high-risk disease and low-risk disease on the pathology, on standard H&E images. And this was a breakthrough that my postdoc George Lee made a couple of years ago, where he was looking at these tumors and he found that when you looked at the glands within these prostate specimens, if you look at indolent disease or low-risk disease, and that's what you see in that right panel, the glands all line up in a very ordered, coherent way. So they're all sort of lined in this sort of more linear arrangement. But if you look at the more aggressive disease in the left panel there, you see the glands are sort of all over the place. And this indeed was actually the original inspiration for the collage idea, the gradient orientation features that I talked about and I presented. That idea actually came from here because we saw in the prostates on the histology that the gland orientations were fundamentally different between high risk and low risk disease. So this is where we sort of applied this notion of entropy to look at the gland orientations to capture the order or disorder between the gland orientations, and use the entropy idea to do a couple of things. One, distinguish between patients with low-risk disease versus high-risk disease, but also just to see whether we can use it to predict cancer presence. And this was published in PLOS One a few months ago, and we had about a 76% accuracy in predicting biochemical recurrence. And just to put that in context, 76% is actually way better than a number of the state-of-the-art approaches that is used in the clinic today. So the Catan nomogram is the de facto way of trying to predict outcome in prostate cancer patients. So the 76% did way better than the Catan nomogram. What was also interesting is when we combined the features with stage, and that's sort of that Kaplan-Meier curve up in the extreme right panel there. It was like black and white. We could separate out the biochemical recurrence from non-recurrence patients almost perfectly. So this is very, very exciting. I know I'm running out of time. Just a couple of more examples or use cases that I want to highlight. I talked about fusion, so I'd be remiss if I didn't actually give one example of fusion of molecular data and pathology data. This is work we just published in TMI, or it's in press or just come out in TMI, where we looked at a series of grade matched and stage matched prostate cancers. So this is particularly difficult because pathologically, or to a pathologist, these tumors look identical. So we looked at these tumors and said, in these tumors that look identical to a pathologist, where the Gleason grades are the same, where the stage is the same, can we combine molecular data and histomorphometric data to come up with a better predictor of biochemical recurrence? So in these patients, we got mass spectrometry information from the dominant nodule. We digitized the images and looked at the histomorphometric features and created Kaplan-Meier curves based off the histomorphometric features alone, the proteomic molecular features alone, and then combined the proteomic and the histomorphometric features and created a fused predictor. And these are the results that we got and were showcased in the paper. So upper left is the histomorphometric features. You don't see the separation, and that's understandable because these are grade matched and stage matched. So there wasn't enough information there. The proteomic features did marginally better, but not statistically significantly able to separate out the two categories of tumors. But when we combined the proteomics and the histomorphometric features, then it turned out we were actually able to separate out biochemical recurrence from non-recurrence in a statistically significant way. George has also been sort of taking this idea to explore the contribution of benign regions in predicting tumor recurrence. So you know, like I said, we tend to focus only on the tumor regions, but what about looking at other areas that are benign, that are normal? Are there features there, are patterns there that could tell us about outcome of these patients? So can we combine the features within the tumor, but also look at other areas within which are normal and see if there are patterns there which combined with the tumor features could allow us to predict outcome? And this, again, was an abstract that was accepted for the USCAP meeting that we'll be presenting in March, where sort of this notion of field effect, that's what it's called, where we combine the features from the adjacent normal regions with the tumor areas to be able to build better predictors of biochemical recurrence. So this was very, very exciting, and we've done this in about 80 patients from Johns Hopkins. Let me leave you with the very, very last use case on lung cancers. And this is work we just presented today in Chicago at the RSNA meeting, just literally 5 hours ago. This is a problem in lung cancers that on CT, there's not much information there. I mean, this is really what you see on a CT for a ground glass nodule. So this is a common type of lung cancer that you see, but that's a blob of gray. That's all you see on a CT for a lung nodule. But there's a big interest now in trying to figure out the extent of invasion within this ground glass nodule, because the extent of invasion has significant implications for the outcome of these patients. So you could have essentially no invasion, which means that outcome is excellent. You may not even want to take out this ground glass nodule. You may have minimal invasion, which makes this patient a candidate for active surveillance, or you have frank invasion, which means you need to resect this nodule out immediately. But that's all a radiologist sees on a CT. So what happens is you just wait, and you just have the patient come back and do a CT scan after CT scan, and you look at it, and if it's growing, at some point you say, oh, it's grown quite a bit, now we got to go and take it out. But by that time, you don't know what's happened. You don't know whether there's metastasis that has occurred, whether it's spread. So we were trying to address the question of, if you have pathology already, if you go back and look at patients who've had nodules resected, could we fuse the pathology with the CT, the preoperative CT, and now map on the CT the regions of invasion, and now tease out, using texture and using image analysis, what are the signatures for the in situ compartments and the invasive compartments, and therefore start to build better predictors for identifying the extent of invasion on the ground glass nodules. So this again was done by two of my postdocs, Mirabella Rusu and Madhi Oruji, who looked at a series of patients where we had the nodule resections, we had the pathology that was digitized, we had a pathologist go in and map out the in situ compartment and the invasion. What we then did was created a histological reconstruction of the individual pathology slices, created that in 3D, and then did a 3D registration between the reconstructed histopathology and the preoperative CT, and that allowed us to now map the in situ and invasive compartments onto the CT, and therefore tease out what are the texture features that can now separate out the in situ compartments from the invasive compartments and move towards quantifying on the CT alone the extent of invasion. So this is the data that we just presented at RSNA. In about 29 patients and had an AUC of in the mid-80s, about 85%, and separating out nodules that had frank invasion from those that had minimal or no invasion. So this notion of radiology pathology fusion we're bringing to bear on multiple different use cases. Another one that I won't talk about, but I'll just throw out there, is in colorectal cancers, where we're using this notion of rad path convergence to try to assess the effect of chemoradiation and learn image features to try to figure out what the effects of chemoradiation are. So let me just end there and just leave you with some concluding thoughts. So I think hopefully one of the messages you get from this talk is that analytics, be it in digital pathology or radiology, really can play a very important role in enhancing our understanding of disease aggressiveness, and more particularly being able to figure out who to treat, how to treat, and really ask questions about treatment response. The image analytics on digital pathology can really help enrich the imaging and molecular data, and hopefully that message came across. But it's very important to understand the key for analytics is really large, well-annotated data sets with imaging and pathology and associated clinical outcomes. So that's a key ingredient. And what I've also not mentioned here, and an important message for us engineers, is the ability to learn, to work with, to speak to clinicians, and sort of get on the same page. Too often we get trapped in thinking about just algorithms and thinking about new methods and comparing it with ABC et al. And DICE and TRE and so on. And these are important things. But it's also very important not to lose sight of what the clinical problem is, not to lose sight of the fact that at the end of the day, if we're doing this sort of work, we need to make sure we include the clinician, include our collaborators, our clinical collaborators, because if you're developing algorithms in a vacuum, then you've got a brilliant algorithm that performs at 97% accuracy that no one will use, right? So it's a very, very important message, if you will, that you need to make sure that you engage your clinicians and clinical collaborators. So just want to acknowledge all my various collaborators at the Case Comprehensive Cancer Center and at Case Western Reserve University, and of course the funding agencies who put food on the table for us and our group, and of course most important of all, my group. I just like to come in and take credit for all the work, but really these are the superstars, these are the guys who do all the work. So let me just end there, and thank you very much.
Time for a couple of questions.
And sorry for going over. So, yes. Yeah.
Thank you very much for your talk. I found it interesting how you focus on subvisual features. I
Yep.
assume clinicians use visual features
Right.
because they can't see subvisual features.
Correct. Correct.
And so why the distinction, and why pursue subvisual as opposed to use the metrics which clinicians are already comfortable
Yeah, because I think there's a lot of information there that we're leaving on the table. I think there's, and hopefully that came across at least partially, that there are pieces of information there, there's embedded data there that a clinician is not going to be able to see, a radiologist is not going to be able to see, a pathologist is not going to be able to see, but has prognostic implications. And with the right tools, this becomes yet another tool to provide to the clinician. It's important to understand we're not talking about replacing the pathologist, replacing the radiologist. CAD for mammography came out in the early 90s, right? And at that point, there was all this pushback from radiologists, oh, CAD is going to put us out of business. It's been 2 decades, where do we stand? We don't have enough radiologists, right? And it's the same pushback when I talk about this from pathologists. Pathologists saying, oh, this stuff is going to put us out of business. No, the fact is we don't have enough pathologists. We've got 15,000 pathologists for 300 million people in the United States of America. That's not a lot, right? And now you go to India, 1.2 billion people. How many pathologists do you have? 700 pathologists. This is not going to put anyone out of business. The point here is we need more trained physicians, but we need the physicians that we have to be able to work more efficiently. And the hope is that with the right analytic tools, they can go in and prize out information that they can't visually see, or would take too long for them to go in and visually see and visually appreciate, and really help to move things along at a higher throughput. So I think that distinction is important, but that distinction is not one that is meant to intimidate the physician. It's certainly not meant to suggest that subvisual features, because you can't see it, means that you're not going to have a job tomorrow. In fact, it's a complement, it's an add-on, it's a decision support. Yeah. Yeah.
When you go from histology to some radiological image,
Yeah.
how confident are you that where you were looking at in the histology is going to be the same place that you're looking at in radiology?
Right, right. Yeah, I mean, it's an excellent question, and it's exactly the issues that you're having as you're looking at co-registration problems. I mean, it's all the same challenges. And of course, I've completely glossed over all those issues because I want to sort of convey the bigger picture. But, it requires getting a pathologist and radiologist to sit together and looking at these images laboriously and, trying to find those fiducials, trying to find those landmarks, and trying to do the best we can in assessing target registration error and DICE and so on and so forth. I mean, they're classical, these are the classical problems that we face in evaluating co-registration algorithms. The other challenge, of course, in this particular problem is, when you're doing the sectioning happens independent of the radiologic data. So, very often the challenge is, well, are we even looking at the right slice? Are we looking at the right section? So it's certainly not a perfect science, but the key is, of course, making sure that the right players are at the same table and looking at the data together, right? And that's really what we found is our best bet of trying to come towards some confidence. And there's going to be error. I mean, we know that. There's always going to be error. This is not perfect. But the way I look at it is, well, you've got 2 options. One, you can do it and admit that this is your error and try to quantify the error that best you can. Or you don't do it, and we continue going on the way we do. So, Tim, you had a question? Yeah.
People at Oxford, Mike Brady and company, they've done a lot of this quantitative image processing into mammography. And they swear by phases, image phase and phase symmetry. Have you
mean phase?
Your local gradient orientation measure with what they've done? I'm just curious to see if,
so this is phase. Are they looking at phase of phase of the images? Yeah. So no, we've not done that. And I think it'd be an interesting experiment. I certainly don't mean to suggest that this collage feature is sort of the be-all, end-all. I mean, if anything, I think it probably is a part of the puzzle. Why I am excited, or why I like the collage approach, is that it connects back to intratumoral heterogeneity. Because in fact, the brain surgeon, the neurosurgeon we work with, gets excited because one of the key problems with brain tumors, I guess brain tumors just tend to be way more heterogeneous than a lot of other tumors, is that very often when you go in, you don't know where to biopsy, because just moving your needle just slightly over, you may be hitting a part of the tumor that is not reflective of the overall outcome or risk of that tumor. So the way this has been set up is that it provides, because it's sort of more localized, looking at these local orientations, it really is tuned to be able to try to capture that intratumoral heterogeneity. So our neurosurgeon wants to do a clinical trial where he looks at these heat maps and goes in and targets the highly expressing regions and see whether, is that potentially more indicative of the biology or the behavior of the tumor. I got to go back and look at phase and see, if we can do a comparison and see how that contrasts against this. Okay.
Based on a question, I guess we close the session and